this video, I'm going to be talking about quorum sensing. Now, what that is a bacterial communication system using chemical signals to coordinate group behaviors. And it's related to biofilm formation. And the way bacteria do that is that they adhere to surfaces and produce extracellular polymeric substances, or EPS in short. So cancer cells and pathogens, they use biofilms as a way to protect themselves, and they even escape or avoid immune attack, or they shield against immune attack. Like, for example, against macrophages. When macrophages attack cancer cells or pathogens, they use a phagocytosis, which is like an engulfing sort of mechanism or action. But the macrophage cannot do that if the biofilm is shielding and protecting the cancer cell or pathogen. So I will name off a list of foods that can help degrade and get rid of biofilms so that the macrophages and T cells can attack cancer cells and pathogens. So the first one here is going to be pears. Pears are rich in fiber, and they can even feed the gut bacteria. So pears, they contain and are rich in chlorogenic acid, arbutin, and flavonoids. And these polyphenols can inhibit quorum sensing signaling molecules in gram-negative bacteria. These molecules are called lactones. And as a result, pears reduce biofilm formation and bacterial virulence. So another thing that triggers quorum sensing, which triggers a biofilm maturation, is bacterial stress signals. But antioxidants in pears can reduce these bacterial stress signals. The next fruit here on the list is going to be peaches. So they also contain polyphenols and flavonoids and phytochemicals. And just like pears, they can inhibit quorum sensing signals from lactones in gram-negative bacteria, interfering with quorum sensing. As a result, reducing bacterial coordination, lowering pathogen numbers, and altering microbial community dynamics, because that's basically what biofilms are. They're like these huge communities of bacteria working together. So peaches, they can make bacteria more susceptible to clearance by shifting the gut or environmental microbiome toward a less adherent and more planktonic state. So another great and important benefit of reducing or clearing a biofilm forming bacteria is that this can favor saccharolytic species, and these species, they are fiber fermenting bacteria. So that means that you can get the benefit from high fiber foods if you eat them, so that the saccharolytic species can ferment the fibers. But the saccharolytic species, they can't do this if there are too many biofilm forming bacteria. So peaches, they support the beneficial bacteria, or and at the same time suppressing the pathogenic biofilm forming bacteria. And it can also reduce the TMAO producing species. So basically, that's TMAO can lead to heart issues. Okay, the next compound or food here is oranges, and the key compounds are hesperidin and naringenin, and these are flavanols that can actually block the lactones used by gram-negative bacteria. Uh, these orange compounds, they also bind quorum sensing regulators, which are LAS-R and LUX-R type receptors without activating them, therefore reducing the expression of quorum-sensing controlled genes at the chromatin level. Inside the chromatin is the DNA, and that is how the genes are controlled inside chromatin. As a result, oranges reduces virulence factor production, reduces coordinated bacterial behavior, and the main goal, reducing biofilm maturation. This way, macrophages can engulf cancer cells and pathogens. There have been observed effects of orange flavonoids when it comes to bacteria such as E. coli and also Pseudomonas. Because of the weaker early biofilm attachment and making it increased susceptibility of biofilms to host immunity and antimicrobials. The master regulator of biofilms is called Cdi-gmp, which is short for cyclic diguanylate monophosphate, and it is a bacterial messenger, and it's found in bacteria and regulates biofilm colonies, also regulates virulence factors. And Cdi-GMP can actually affect cancer indirectly, because what happens in humans, Cdi-GMP is recognized as a foreign bacterial molecule by the immune system, and then activating the STING pathway, short for stimulator of interferon genes. And when immune cells detect Cdi-GMP, STING is activated, type 1 interferons are produced, and then anti-tumor immune responses increase. Okay, chronic biofilm infections can lead to persistent inflammation. So that could attract cancer cells, because cancer is attracted to environments of high inflammation. So biofilms are not good. Also immune dysregulation, and chronic biofilms can even damage DNA. That could have an effect on gene transcription factors, therefore increased cancer risk. Some examples, and this links back to my the oranges that I was talking about earlier, is H. pylori gastric cancer. So it's critical to reduce biofilm formation. So H. pylori bacteria can actually, they use biofilm related mechanisms regulated partly by Cdi-GMP in many species. And emerging research also shows that tumors, they contain bacterial biofilm-like communities, and these tumor biofilm communities are especially present in colon cancers and pancreatic cancers. But this is still an active research area. Okay, so quick recap: pears contain flavonoids that reduce biofilms, also fiber to feed gut bacteria. Peaches also do the same thing, as well as oranges. So they all reduce the bacteria that produce the biofilms, and allowing the fiber fermenting bacteria to also thrive as well, so that dietary fiber can be fermented by those bacteria. The master regulator of biofilm formation is Cdi-GMP. A chronic biofilm formation can lead to high inflammation, which cancer is attracted to, and even DNA damage, which is also another cause of cancer. If biofilms are not degraded, the macrophages cannot engulf or get rid of cancer cells and pathogens if they are protected by biofilms. So that's it for the video. Like, share, comment if you want more future videos, and I'll see you next time.